Tonight, as Peter said, we are going to hear about the development of a new drug, a world-first drug that dissolves plaque, the type that builds up in arteries. We're not talking about teeth at all. It was a cancer researcher working out ways to regulate the massive blood vessels that sometimes grow around some tumours that bring more nutrients into the tumour, who made the discovery. And that was here at the Harry Perkins Institute of Medical Research. And because of that discovery, that researcher has pivoted her career from cancer research to cardiovascular disease. And of course, that researcher is Associate Professor Juliana Hamza, in the green, two along from me, Head of Laboratory of Targeted Drug Delivery, Imaging and Therapy here at the Perkins. For tonight's discussion, Juliana will be joined by, on my immediate right, Mr. John Barrington, AM, co-founder and CEO of Artery Limited, a healthcare company investigating the use of artificial intelligence to improve diagnoses of coronary disease through the detection of plaque. And John is also the chair of the Harry Perkins Institute. Next to John is Professor Shirley Jansen, Head of Vascular Surgery at Sir Charles Gardner Hospital and Director of the Heart and Vascular Research Institute here at the Perkins. Juliana is on Shirley's right, and then at the end we have Mr. Trevor Jones, very generously giving his time, being a poster boy for us tonight. Trevor is an amputee resulting from both his diabetes and the build-up of plaque causing vascular problems that contributed to several lower limb amputations. And thank you, Trevor. We are really grateful for you being with us this evening. Juliana, I want to start with you. Can you remember the moment, if it was such a thing as a moment, when you really recognised this drug you were working on for cancer was having an effect dissolving plaque?
It starts about 7 years ago, Miriam. We knew at that time that it will have an effect in the large blood vessels, but we were still very surprised to see the extent of the treatment outcomes when we see a very effective and safe plaque removal from the blood vessels.
And surely, I believe Juliana was actually not as measured as that, but ran down to the cafe to tell you all about it.
She did. I was having lunch, and I remember Juliana running in with a picture of some slides, and there was a group of 10 blood vessels at the top with one colour and 10 at the bottom with another colour, and the red meant there was plaque, and there was no plaque in the ones at the bottom. And once she'd explained it to me, it was really exciting.
Juliana, it must have been quite a moment. Can you set the scene for us a little bit and remind us or describe from a scientist's perspective what plaque is, what plaque looks like when you're looking at it?
Well, plaque is actually the scar tissue that forms and grows in our vascular wall. It consists of high content of lipids and cholesterol deposits and also calcification around the vessels. Now, we probably need to understand a bit more about what happened in our blood vessels. We know that our blood vessels supplies oxygen and nutrients to different parts of our body from the heart. And we need to think of it like garden hose to water the plant, really. And in a similar form, our blood vessels is very flexible, very elastic, and it consists of this extensive network of cells that contract all the time to allow the vessels to expand and narrow so that it can accommodate blood flows under different pressure. So the problem is that because of the wear and tear as we grow older, and the vessels start to lose flexibility over time. Now, this is a major problem when we have high cholesterol and lipid content in the body that absorb in the cellular component in the blood vessels. Once the cells in the vessel wall absorb cholesterol, they become weaker. And this means that they start to secrete more collagen and scar tissue around them. And this is what creating, hardened plaques that consisting of encapsulated lipid engulfed in these scar tissues. And this can cause a major problem because plaque can happen in any types of blood vessels.
So, Shirley, as a vascular surgeon, how significant could a drug that dissolves plaque be in your world?
Oh, highly significant, Miriam. At the moment, all we can do is treat the end effects of vascular disease. So if it's in the arteries to the heart, that's a heart attack. If it's the arteries to the brain, that's a stroke. If it's the arteries to the leg, then obviously someone like Trevor, who has lost his leg for that reason. So at the moment, all we can do is treat those when they happen. We can't prevent or cure the narrowing in the arteries from plaque. A lot of the medicines that we use today are really just to control the risk factors for vascular disease that I think we'll come to a bit later. But at the moment, we've got nothing to cure the plaque. And so this could be a game changer.
Yeah, and how commonly, is this a disease of ageing? Do we all develop plaque as we get older?
We do. We all develop it as we get older, and there are some genetics involved, of course. But the more risk factors you have, the worse the problem is. And those risk factors are smoking, diabetes, blood pressure, high cholesterol, being overweight and having an unhealthy lifestyle. So the more of those factors you have, the more your risk ramps up.
Mm. Now, John, you're not actually working with Juliana and Shirley. You're working on another project, a company, Artery, which is working on reducing the risk of built-up plaque by detecting it with AI. Can you briefly describe in language that we understand what Artery is working on?
So I won't be talking about convolutional neural networks in detail. But we're earlier in the whole process, very complementary to what Juliana and Shirley are doing. We're attempting to detect the vulnerable plaque that Peter mentioned before it becomes a problem. So it's about screening. There's bowel cancer screening now, mammograms, of course. There's not screening for heart disease, and yet we know that if the vulnerable plaque is in the arteries, there is a significant increased risk of heart attack and death. What we're attempting to do with where we are doing now with artificial intelligence is detecting this soft, vulnerable plaque, which is inordinately difficult to see with the naked eye.
So how do you use AI? How does it work?
The machine is really looking at pixels from a CCTA scan, a CT scan of your heart, and I'm sure many people in the audience have had a CT scan, rather than a radiographer and a radiologist taking hours to days to interpret that scan and report. The artificial intelligence can assess the CT scan within minutes, develop a 3D model which identifies the location and severity of the disease, and generate a report for the radiologist and cardiologist to use to prescribe drugs, recommend lifestyle changes, whatever it might be.
So without the AI, the type of identifying of plaque that we're talking about or hoping for could take how long?
It takes a significant period of time. The radiologists or cardiologists to detect the vulnerable plaque have to be level 3 readers, as they're known. They have to be expert readers. They have to be trained in this. And because it is so difficult to see, it takes potentially hours to generate the report. In fact, there was 250,000 people in the United Kingdom waited a month for their standard CCTA report. And of course, heart disease has a time criticality about it. The vulnerable plaque is not generally reported because it's so difficult to see, and because there are not a lot of people trained to identify the vulnerable plaque. So we're attempting to address that. One, reporting it hasn't been generally reported, and two, to do it very quickly.
And how significant is this vulnerable plaque or dangerous plaque in the story of cardiovascular disease?
There's significant research out of Scotland, ScotArt, which is a significant study in Scotland late last year. Michelle Williams led that study and found even a plaque burden of as little as 4% increases the risk of fatal and non-fatal heart attack by a factor of 5. So this is significant. And the issue is that we haven't typically reported this. As I say, we're reporting indicators, biomarkers that are not the real cause of heart attack and death. So Juliana's research and with the drug to eliminate that plaque is a significant step forward beyond the identification.
Shirley, how are you actually working? Because you're over at Charlie Gardner's as a vascular surgeon. Juliana's here in the laboratory. How are you actually working with Juliana on this journey?
So I consent my patients who are having open surgery where we remove plaques for various reasons. And usually that plaque is just disposed of. It's not needed for anything. We don't need to analyse it in the hospital setting. And so patients are always very willing to donate that plaque for research. So I then take it from the operating theatre, bring it round here to the Harry Perkins next door and give it to Juliana straight away so she can run tests on it and examine it and test it against her new drug.
And do you see, Juliana, different sizes in plaque, different look, different patient, if someone who's been on medication or not? Do you see a whole range of variation?
Yes, very. We have collected over 90 samples from the patients, and they all varies. Even in the same vascular bed, you can see a different composition. And this is what I think a problem with the plaque stability. But we also find the more unstable they are, the more lipid content that accumulate in the wall of the vessels.
Now, I guess Shirley, I'll come back to you. In the drug development story, I'm sure I was like probably most people in the audience. I thought we were going to then talk about the development of this drug for heart disease or cardiovascular disease and stroke. But actually, the pair of you are focusing this development of the drug in relation to another disease called Peripheral Arterial Disease, or PAD, P-A-D. Was that for a strategic reason that you're developing the drug in relation to PAD, not heart disease first up?
It was because the amount of research in the heart space around heart attack and coronary artery disease is huge. And quite appropriately so, but it means as new researchers it's very competitive to get funding. Peripheral arterial disease, or PAD, is a very similar condition. It's associated with plaque buildup. It's the same kind of plaque. It's cholesterol laden. It causes narrowings and blockages in arteries. And so it's a more descriptive concept really of the whole process, which includes heart disease, includes stroke, it includes problems in the legs such as that Trevor has had. And so that was partly strategic because we want to bring this drug to market as soon as we can. So we've got a lot of testing to do and safety regulations, etc., which you'll hear more about later. But in order to speed up that process and secure funding, we chose to apply it to the peripheral arterial disease space.
And Juliana, you were telling me it was also about developing a drug for more of an unmet need.
Yeah, I think we also need to think about practicality and strategic decision because with the hard research, for drug development, it takes a lot longer, more sophisticated readout to look at the improvement in health outcomes because we can't predict when the patients will have heart failure. So the duration of the trial will be much longer. So it's more costly and takes more time and larger number of patients. But when we talk about peripheral arterial disease, it's a huge clinically unmet need, and according to Shirley, that it's a more straightforward and more primary outcomes that we can measure is a kind of more immediate, isn't it, Shirley? That's why.
Because we can measure wound healing and we can look at blood flow in the limb quite easily, and so we can see firsthand if that's not going well.
How prevalent is PAD?
So PAD is really prevalent. It's an underestimated burden, as Juliana has just mentioned. About 220 million people globally are suffering with peripheral arterial disease. And that equates to about one in 5 West Australians who have peripheral arterial disease. And the healthcare costs, the burden is huge. So in Australia, we spend about $1.5 billion on the healthcare costs associated with peripheral arterial disease.
And the symptoms?
So the symptoms usually start with a cramping pain in the legs on walking. That's the first sign. We call that claudication, and that's the first sign that there are a limitation to the blood supply to the calf muscles when you walk. And as that progresses, that pain happens earlier and earlier until when it's happening at rest, that's a sign that the resting tissues, the resting muscles, don't have enough blood supply to survive. And that's really when a limb threat situation is present. And that's when we need to look at doing angioplasties, stents or bypasses.
And Trevor, you've had a really long and difficult journey, contributed to having diabetes, but also peripheral artery disease. The impact is obviously quite visible. Yes, can you walk us through? That was really unfortunate.
I can walk you through it. It started with me in 2018. I had a infection in my big right toe on my right foot, which was the first amputation. Then trying to look after the rest of the toes. The second toe got an infection in it. One morning I was drying my toes after having a shower, and a big lump of white skin dropped out of the end of the toe. So straight on the phone to my GP, and he said straight to the hospital. He could see the end of the bone. So therefore another operation was scheduled. So that toe was removed, and then there was a third operation to remove the other three toes on my right foot. So I have no toes on the right foot. I had a remnant part of the second toe that was sticking out that actually ended up getting infected and created a few problems that I had to go in and have other operations. Then we had the left ankle developed a diabetic ulcer, in which they cut a hunk of skin out about that square in a triangle, which never healed. So one day in August of 2018, I saw the surgeon and he said, we're going to cut your leg off above the ankle. I said, no, you're not. And went away and had to go back and see the surgeon again, and ended up going into hospital and having a below-the-knee amputation. The first amputation didn't heal all that well, and some 9 months after that operation, Professor Jansen did another operation, shortening my limb by another 50 mils and bevelling the tibia back at 45 degrees, stitching the suture line, which is healed correctly, and I was then able to do all the rehab, etc., to get back to walking.
Mm. And Shirley, what other treatments would be available for Trevor?
Well, the medical management, which we touched on earlier, is really aimed to reduce risk factors for recurrent disease. It's not going to treat disease that's already there, and so that's the blood pressure lowering, anti-diabetic drugs, stopping smoking, losing weight, controlling blood pressure, etc. But they won't really halt the disease process that was happening in Trevor, and that was really worrying. And diabetic patients are more prone to infection, and infection and poor blood supply is a really bad combination. And so a limb threat can really happen very rapidly, and we need to get control by removing all the dead tissue. So aside from the medical treatments, we can do angioplasties and stents, and I think Trevor had some of those as well, and those are really aimed to just squeeze the plaque and cholesterol back against the wall. So we've got a new channel. Sometimes that needs holding open with a stent, just as we do in the heart. And if that doesn't work, we can stitch in a bypass. But none of those treatments actually cure the problem. They don't remove any plaque, they don't dissolve it. And so Juliana's drug hopefully will be a new first-in-class game changer.
So there is no other drug that dissolves plaque, is there? No. Juliana, what is this drug? What's it made of?
Well, it is actually a protein that's made in our body when we are in stress in particular. And but in high amount it's very toxic. So we produce them in the lab and we modify it. we attach the drug with a small molecules that can specifically recognize the scar tissue that I'm talking about in the atherosclerotic plaque. The idea is that we can deliver this drug more specifically to target the specific tissue that we want to treat, and that can remove the side effects associated with the non-specific binding of drugs in different tissues.
So how do you actually specifically deliver a drug to exactly where you want it to go? Because you're saying that at high concentrations, this is a toxic drug, even though it's something that we produce under stress naturally. How do you actually get it to where you want it to go?
We call this approach a targeted drug delivery approach. We think of it like a postal delivery service. So every part of our body contains specific codes that we can identify molecules that bind to these codes. Now, plaques is the same. It has specific codes. We found the molecules that bind to it. So what we did is that we permanently attach this drug to this code, molecules that bind to specific codes. So when it circulates, available in the blood, it will allow a specific accumulation that sends where the code is in the plaque tissue. And that's the way that we can improve concentration and reduce side effects.
John, clearly there's great reason to commercialise this drug, but the road to commercialising any medical research is long and expensive. What sort of time trajectory might this development take?
In the drug arena, it's potentially 10 to 15 plus years. The word I thought of when you were thinking about describing it was arduous. It is long and arduous. It's very difficult. There will be the multi-phase clinical trials. There will be the regulatory approval processes. And so persistence is an important attribute of anybody developing a new drug or a new technology. We're in a slightly different space. It is shorter time frame for medical technology than pharmaceutical space, but similar challenges. And
I mean, how much, in the 3 years of Artria, give us a sense of how much investment, what do you need to build around you in a team to succeed? In your 3 years, what's happened?
We've raised $60 million. Artria is a West Australian company headquartered in West Perth. We have people in Perth, Melbourne, Sydney, London, Los Angeles, Washington DC, New York and Phoenix, Arizona now. So it's a global company out of West Perth. We listed on the Australian Securities Exchange in November last year. As I said, we've raised some $60 million. We have a team of 30-odd people in West Perth, plus those other people around the world. And the important thing, I think, is having the talent around the developer, the scientist, and it's multifaceted talent. I think that's the most important part of it, because it takes so much and so many complementary skills to bring something to market eventually, whether it be a drug or a medical technology.
And obviously investment. I mean, right now, what sort of investment does Juliana need?
Juliana's probably better to talk about that, but we have chatted about it, and the next phase you're talking multiple millions of dollars in the next phase. It always takes twice as long and twice as much money. So that's only the next phase, isn't it? So then from there, the costs ramp up significantly. And the unfortunate thing, this is all about, the parallel between what we're doing, it's all about saving lives. This is the largest cause of death in the world. So we're trying to solve for that. So the benefit to society is profound. It actually needs a very sound business model. and commercial underpinning to enable that societal benefit to be affected. And there is a complementarity there that without that, it's going to fail.
And does Western Australia have any particular benefits or disadvantages in this? You know, we're a very resource rich, focused society, but we're also everyone seems to know each other. We're a network.
That's an excellent point. There is a very good book, it's called Startup Nation. It's based on good research out of Israel. Why is Israel such a leader in many areas of innovation, medical research and technology? One of the hypotheses around that is because people serve in the national service, the 2 years they do in the national service. They build big networks, and while one person might be a competitor to another, they're all about building Israel. And that network is, as I say, a suggestion why Israel is so successful in developing technology, because people know each other. If you think of one other city in the world or big town, what would be more networked than Perth? So we have a very, very strong network here, I think. We don't capitalise on it.
And when you think of job prospects, you know, in commercialising medical research, patent lawyers, tax accountants, regulatory experts, clinical trial specialists, the list goes on and on. I mean, the range of people you're working with, from, you know, Professor of Cardiology Girish Dwivedi, who's with the Perkins, all the way through to those AI tech people you've got, plus all the IP. The jobs are very real for our economy, and yet Western Australia, I think, invests less than some other states in this area, doesn't it?
It does. Just to talk about the talent, all of our talent, there's 14 nations represented in our group, and yet they're all out of Perth. They've come here to live and they've come to stay because of the lifestyle and what Perth offers. So there's great talent there. And just one other comment on that, one of the downsides, what we're competing against, is the resource sector, who put a lot of money to hiring people. Some of our staff have been offered 40-50% increase in salaries to go to work for the mining companies. One of the benefits we have is we're about saving lives. So that's a good employee value proposition. So the talent is here. There's certainly more that is required, and Minister Stephen Dawson, the Innovation Minister in Western Australia, and also Minister for Medical Research and ICT, has come to the portfolio with great energy. He's done some very good work in a short period of time. He's created a collaboration with NCAD, the globally renowned university. Part of the MBA program there will be a case study out of Western Australia in innovation and technology development. Wonderful. Where we lag by orders of magnitude is the government funding. The Future Health Research Investment Fund, FARHI, we're talking tens of millions of dollars. In Victoria, the commitment to innovation by the state government is $1.7 billion. New South Wales, $1.6 billion, and in Queensland, $775 million is committed to innovation, to funding the science and the development and the commercialisation. Western Australia still lags significantly in that funding arena.
Trevor, when you think about a drug that could dissolve plaque for patients like you, how does that make you feel?
If I'd have had this drug 5 years ago, I wouldn't be sitting here now. Very much so. I would have 2 legs. I would be walking on 2 legs, not a prosthetic. I would probably have all my toes. That's how big I think that this drug's going to be.
But I know you do talk about life after amputation. You're an incredibly positive person, and I don't know if anyone's noticed. Will you describe what you've got on your prosthesis? What are those images?
The Marvel characters. Now, at the time that my prosthesis wanted to make my socket, she said, "What do you want on it?" And she came out with these materials, mermaid scales, purple unicorns, purple fabric, and the Marvel characters. So I said, the Marvel characters. The one thing that I do want is, I want the Marvel sign on the outside of my left leg so that people can see it. I'd never watched a Marvel movie until I got my Marvel leg. I think I've watched 29 of them now. So, and the first one was the best one, Captain America. So,
But Trevor, I don't think really anyone who hasn't gone through an amputation can begin to imagine what it would feel like to be told the news that it's going to happen or to wake up after the operation. How did your mental health cope in those days afterwards?
went through the floor. Went through the floor. I asked the hospital staff to see a psychologist or a psychiatrist. I got nothing. Being an ex-Defence Force member, I phoned Open Arms, who linked me up with a psychologist who I see every fortnight to cut for the current day, who's helped me along the way tremendously. I think that's one thing that's lacking from the hospital side because it affects you tremendously. I had a gentleman by the name of John rock up with a bag of, I call them goodies, from Limbs for Life. Sat down and talked to me for about an hour and a half. Basically, I started reading the literature. I joined Limbs for Life. I've become a peer support volunteer for Limbs for Life, so when somebody has an operation or is going for an operation and want to talk to somebody of my age, not necessarily the younger people want to talk to the older ones, but I can relate to them. I can relate what they're going to go through. But we do say things like there's life after amputation. We learn to adapt. We learn to do things differently.
Yes, how long did it take you before you walked again?
383 days.
Wow.
Excuse me. The first operation that I had to have the amputation did not heal correctly, so therefore it got infected. And as I said before, Professor Jansen had to go in and do the second operation. So basically for most of 383 days I was in a wheelchair. And the day that they actually said ditch the wheelchair and get up and walk, it's a marvellous feeling that you can actually go and do it.
Yeah,
I still have to go around supermarkets holding onto a trolley. I use a cane, but I can walk around the house at home without a cane. Sometimes a bit wobbly, but I get there in the end.
Juliana, you recently hosted Trevor in the laboratory, and to put a bit of context around that, scientists don't often have patients that link. The link might be someone like Shirley who's bringing the patient tissue to you, but you hosted Trevor in the laboratory. Does it make some sort of a difference as a scientist having a direct link with a patient?
Yes, of course, because my team and I were so thrilled, firstly, to have Trevor, and then we were like, oh, wow, to learn. Trevor told us that he has over 18 medical procedures within 5 years, and that really signifies the problems, and we could really looked from the experience that Trevor had to really think about how we can solve some of the challenges that comes up with this drug development. And we really want to make this as part of the consumer or community-driven health problems, and by viewing their concerns and needs of the patients.